All right, I want to finish talking about animal biodiversity. And we talked about animals are far more than just the cute, fuzzy mammals, like the cat that was sitting on my shoulder a few minutes ago, but in fact include insects and spiders and clams and snails and slugs and squid and jellyfish and corals and Chinorhynchs, Loriciferans, and tardigrades, and about 30 phyla in all. And they're all multicellular. They all show cell differentiation. A sponge might have 10 different cell types. You've got roughly 200. Generally, they are heterotrophic, which means that they get both energy and nutrients by eating other organisms. You eat plants and animals and maybe the occasional fungus if you like, mushroom soup or something, to get both the energy and the raw materials to power your own metabolism. Most animals can move at least some stage of the life cycle, although there are some that as adults don't move, corals, for example. Usually the bodies are symmetrical in some way. There are no cell walls, but the cells are held together by a fiber meshwork called the extracellular matrix. This is what enables you to be flexible. The reason that that your face is not as hard as a piece of wood is because your cells are not locked together by cell walls. They're held by a network of stretchy fibers. That's the extracellular matrix. And then all animals except the very simplest, the sponges, have their cells organized into groupings of cells called tissues. Commonly, tissues are shaped like sheets, although some of them are three-dimensional masses. Okay. Tissues are usually sheets, sometimes three-dimensional masses. They're all mostly the same cell type, and they're specialized for one function. And there's 4 basic tissue types in a typical animal body. Some of the very simplest animals, like jellyfish, do things a little bit more simply. But as for you and almost all animals on the planet, you've got 4 basic tissue types. Epithelial tissue, epi means on top of, and these are tissue that lines cavities, or for that matter, is exposed to the outside. Your skin is made up of epithelial tissue, but so is the lining of your mouth. Don't actually do it at the moment, but if you were to stick your finger in your mouth and touch the inside of your cheek, you'd be touching a type of epithelial tissue. If you were to stick your finger up your nose, you would be touching epithelial tissue and boogers. The lining of your lungs and your trachea, your windpipe, the lining of your digestive tract, mouth, esophagus, stomach, intestines, etc. The lining of the tubes inside whatever excretory and reproductive organs you happen to have, that's all epithelial tissue, tissue that lines. Connective tissues are used for structural support and integrity. Bone is living tissue. Not everybody's aware of this, but your bones are in fact alive when you are. They've got lots of living cells in them. They need a lot of blood. They need a lot of blood vessels to keep them alive. I've been told that orthopedic surgery is the bloodiest field of surgery to specialize in because bone has a lot of blood in it when you start sawing it. cartilage is a connective tissue. The flexible stuff in your nose and your ears, and also in your joints. Tendons like the Achilles tendon at the back of your heel. Blood is technically a connective tissue. All of those are connective. And then nervous tissue is used in transmitting signals. So your brain is made of nervous tissue, so is your spinal cord and all of your nerves. And then muscle tissue contracts. So your muscles are mostly made of muscle tissue, duh. But you've got layers of muscle in places like your digestive tract. What forces food from your mouth down into your stomach and on through the intestine is the contractions of a muscle layer outside the epithelial layer that makes up your digestive tract. Most of your heart is made of muscle. You know, your tongue is mostly made of muscle. Right, and so on. Oh, I forgot to mention, epithelial tissue is often modified for secretion. So you have epithelial tissue lining the inside of your mouth, and then you've got a couple of very tightly folded up little pouches made of epithelial tissue that secrete your saliva. That's your salivary glands. Those are made of epithelial tissue, and they secrete your saliva. Mostly water with a little mucus and some proteins we'll talk about later. Just a sample here. This is a slice through a human fallopian tube looked at under the microscope. These are the tubes that convey eggs from the ovaries to the uterus. What you're focusing on here is most of the pink stuff that you're seeing is made of cells that are shaped like very tall rectangles. You can see nuclei in many of them. That's epithelial tissue. It's a type called columnar epithelium because the cells are shaped like columns. It's a little bit different from the epithelial tissue that makes up your skin or the inside of your mouth. But it's epithelial tissue. It's a lining. That happens to be a piece of human cartilage, and the white and the grainy spots that you see, those are the actual cells that make up cartilage. And all of the pink stuff that's surrounding them, that is extracellular matrix. That's what gives cartilage its toughness. The cartilage under the skin of your nose and under the skin of your ears is relatively flexible. You've got a much tougher form of cartilage making up your joint surfaces. You know, where you have a joint, your bones are not actually rubbing on other bones unless you have arthritis, and that's very painful. Your bones are covered at the ends by caps made out of cartilage, and that's what rubs up against each other when you move a joint. And they're lubricated by some fluid to give you easy and pain-free motion, unless the cartilage starts wearing away, in which case you have arthritis. But that's a much more firm type of cartilage. And what makes some cartilage flexible and others firm is all that extracellular matrix, all of the pink stuff that you see surrounding the cells. That's if you want a piece of my mind, there it is. That was a joke, and that's not actually my brain, but this is what brain tissue looks like under the microscope. The cells have been stained. You can see several cell bodies with nuclei inside. They're kind of cross-shaped or star-shaped, I guess. But connecting each cell to the other cell is a bunch of branching fine filaments called the axons and the dendrites. And that's what actually connects cell to cell and makes it possible for your brain to have the circuitry that it does. So that's fairly typical. Nervous tissue, and that's a piece of muscle from, well, I can't tell what muscle it is, but the nuclei of these cells are the flattened blue spots. And then almost the entirety of the cell body is packed full of specialized proteins. They're making up the fine pink stripes that you see are arrangements of muscle proteins. If you must know, they're called sarcomeres. I won't test you on that, but that's what they're called. And the arrangement of proteins that make up those sarcomeres enables muscle to contract. So all of the muscles that move your limbs, the cells look like this, with some differences that we don't need to get into. Most of your heart looks like this as well. And then there's muscles lining, lining of your intestines, an awful lot of your uterus, if you have one, has muscle in it, which is used to push babies out. Yeah, a lot of places in the body have muscle tissue. In some of the simplest animals, like these corals and jellyfish, the body is just made up of several types of tissue kind of folded around each other. But in most animals, including you, several tissues will form integrated units that we call organs. This happens to be a slice through the wall of your small intestine, sliced, stained, and mounted on a slide to look at through a microscope. The inside of your intestine is up at the top, and you can see there's kind of a crumply looking layer right there. That's the epithelial layer. That's the lining of your small intestine. It lines it, it protects the organ from anything rough in the food that's passing through it. And it's also got lots of little glands that secrete what we call enzymes that break down the food. And then the food gets absorbed into that epithelial tissue as well. And the molecules that made it up get transported to all parts of the body. Anyway, that's roughly the top quarter of this picture. Below it, there's a kind of raggedy looking layer that's connective tissue. And below that, making up most of the thickness that you see, is two muscle layers. There's some more connective tissue on the outside that holds the whole thing together. And you cannot really see them in this picture, but there are nerves that run to your intestine and control its activity, control how fast it contracts, how fast it pushes food through. If you are physically exerting yourselves, it tends to shut down because you need energy for other things than digestion. If you relax, it tends to speed up. If you've got food poisoning, it tends to move very rapidly indeed. So there are nerves in your small intestine, although they're not visible in this picture, but you can actually see the other three tissue types in here. And then if you take several organs and integrate them together into doing something, we call that an organ system. So that's the human digestive system, starting with your mouth and the salivary glands up at top. They're labeled parotid glands, submandibular, and sublingual glands. That's just the glands that make your saliva. You see the esophagus leading to the stomach, emptying into the small intestine. There's your liver and gallbladder that pitch in, and your pancreas, which secretes enzymes that break down your food. Small intestine, large intestine, leading all the way around to the rectum. Rectum damn near killed him. Never mind. Punchline of a very old joke. So that's one organ system, and in human anatomy, there's about 16 organ systems that we recognize. You might have heard of your muscular system, your skeletal system, circulatory system, nervous system, immune system, reproductive system, on and on and on. This is not the course that most health science majors usually take, but if you do go into health science, you get to take anatomy and physiology or structure and function of the human body, and you'll go through all of those systems in agonizing detail. Not something I teach, but I've done it before. We actually taught a class with human cadavers in it that was a lot of fun. Right. And that's all we're going to say about living biodiversity. But one thing about life that I haven't mentioned yet is there's not only a huge amount of living biodiversity, there is a ridiculous amount of dead biodiversity. Okay, sorry about that. Please apologize to Brennan Price for me. Brennan, I'm sorry, I thought I'd already admitted you. Evidently it didn't take.
Oh, you're good.
Okay. By the way, everybody, I just realized that I can't see a clock from where I am at the moment. The clock I usually use seems to have run down, meaning somebody please give me a heads up when we've got about 5 minutes to go so I don't run too far over time, if you don't mind. All right, so yeah, holler when it's about 11:45.
It's 10:30 right now, just
10:30, okay. Okay, 10:45 I meant. All right, okay. So I've said my piece about living biodiversity, but you can't go any further without noting that there is a lot more dead biodiversity, like this Tyrannosaurus Rex skeleton right here, than there is living. And we can study it by looking at fossils. And a fossil is defined as any trace at all of an organism that used to be living and now isn't. Up there at the top, that is the beautifully preserved skeleton of a fish. By the way, that's from Fossil Butte National Monument in Wyoming, just outside a little town called Kemmerer. Worth a visit if you ever get to go up there. They happen to have some beautifully preserved fossils like that one. Below, that's a picture that I took at Clayton Lake State Park in northeast New Mexico. There's a river there. The state built a dam to make a lake, nice place for a swim or water skiing if you like that. But where they scooped up soil and rock to make the dam, they exposed a lot of bare rock. And this is one of the surfaces that was exposed. And in the center, towards the bottom, I don't know if you can see my arrow. I'm circling it now, but sometimes that doesn't get transmitted. But in the center down there, you can see there's what looks like a depression in the rock that's got 3 little projections on it. That's a dinosaur footprint. The other reason to go to Clayton Lake State Park is it's a great place to see dinosaur tracks. Now, the largest trackway of dinosaurs ever was actually found in Saratoga, Arkansas. Little known fact. But that's an example of a trace fossil. It's not an actual body part, but it's a trace of a living thing's activity in the rocks. Incidentally, there's no real cutoff on how old something has to be to qualify as a fossil. The old seashells that you might pick up at the beach, they count. They're fossils. They're not, I mean, they might be just a few days old, but they're still fossils. They are pieces of something that used to be alive and now isn't. If you look in the cement, the concrete on the sidewalk outside the student center, they built that about 25 years ago. And there's a few places where leaves fell and landed in the wet concrete and left impressions. Those are fossils. Only 25-year-old fossils, but fossils nonetheless. Virtually all rocks where you find fossils are sedimentary rocks. A sedimentary rock was formed by either water or occasionally air, depositing layers of mud, silt, or sand. If you were to go down to the mouth of the Mississippi River, for example, the Mississippi dumps something on the order of 250 million tons of mud and sand into the Gulf of Mexico every year. And that stuff eventually settles to the bottom and is building up now on the bottom of the Gulf of Mexico in layers. So those are sedimentary rocks that are forming now. Some sedimentary rocks are deposited by evaporation. If you have a salt lake and that dries up, what it leaves behind is the salt, or by chemical reactions that we don't need to get into, like a type of rock called limestone. There's a lot of limestone up in the Ozarks, for example. So sedimentary rocks are laid down by the action of water or rarely air, and they're laid down in layers. And one of the first people to seriously think about how they formed was a Danish physician back in the 1600s named Nicholas Steno. Incidentally, he later became a Roman Catholic priest, which was dangerous in the 1600s because this is when Europe is fighting the wars of religion, and Catholics in Denmark tended to get attacked by roving bands of evil Lutherans. Long story there. He ended up being beatified in 1987. That's the last step before sainthood. So recognized as a very holy man by the Catholic Church. But he was also one of the first people to think about how fossils and sedimentary rock layers might form. And he came up with a couple of rules that we still use. They're sometimes called Steno's laws, although they're not strictly speaking laws. There are cases where they don't work, but they work very well as guidelines for looking at sedimentary rock layers. Steno's law of superposition says that whenever you have a stack of sedimentary rock layers, the ones on the bottom of your stack are the oldest, and the ones on the top of your stack are the youngest, and the ones on the in-between of your stack are in between. Think about my desk. You can't see it, but I've got a pile of letters, junk mail, and unpaid bills from this month sitting right here beside me on my desk. If I were to move that layer to the side, I'd find a layer of junk mail and unpaid bills from last month. And then underneath that is a layer of junk mail and unpaid bills from the previous month. And then below that, a layer of junk mail and unpaid bills from, what are we, November 2020. And then the October bills are below that, and so on. My mail tends to pile up. And unless I've disturbed the pile by, throwing away the junk mail and paying the bills, the pile is going to build up in a time sequence. The older stuff is on the bottom of the pile and the younger stuff is on the top of the pile. You could test this yourself. If you filled a tub full of water and first you threw in a tub of pebbles, the pebbles would sink to the bottom and they'd make a layer. And then the next day you threw in a tub of sand, the sand would make a layer on top of the pebbles. And then the next day you threw in some mud, the mud would sink down and make a layer on top of the sand, on top of the pebbles. and so on. Those layers would build up in a time order. Steno's law does not say anything about how old a set of rocks are or how long they took to form. On the left, some rocks that I have explored in my traveling days in western Utah. Those are limestone layers in the Drum Mountains. You can see they're layered. We now reckon that they formed roughly 500 million years ago, and it took several million years to form them. On the right, you can see some rock layers on the rim of an extinct volcano called Yuba Hibi Crater in Death Valley National Park, which I have also visited. Those layers formed a few thousand years ago. There would have been Native Americans around to see it at the time if they were dumb enough to get that close. And we estimate they only might have taken a few weeks to form. They're both layered, and Steno's law still applies, and it doesn't matter what the age is or the length of time. This is only a statement of relative age. The rocks on the bottom of that stack on the side of the Drum Mountains are older than the layers at the top. By itself, that's all we can tell. You can get cases where rock layers get tipped and folded after they form, and usually we can tell when this happened. That's on the road cut on Hogan Road, right where they blasted Hogan Road through Cadron Ridge to get to US 64. So if you're going out Hogan north and you pass the new post office and the brick oven pizza and all of those ginormous apartment buildings at Centennial Valley, then you get to the part where they blasted through about 20 years ago to connect Hogan with US 64. So if you go to the dump, maybe you go out that way. If you're going to the dump or if you're going to Morrilton, but I repeat myself. Sorry. Anyway, so I took this picture where they blasted Hogan through Cadron Ridge, and it's made of sedimentary rock layers. The layers are tilted by about 55 degrees. Instead of being horizontal, they're sticking up at a slant. It gets worse if you go to Hot Springs, where the rock layers have been compressed so much that they've folded up. These are on McLeod Street, south of Hot Springs itself. And you can see the layers over at the left are sticking almost straight up, and then they bend down and then bend back up again. We don't need to worry for our purposes as to what did this. It's got to do with what they call continental drift. I'm not going to go into the details further, but suffice it to say that extremely powerful forces in the Earth's crust have caused those rocks to tip and fold and bend and sometimes crack and shatter, making what we call faults. Most of the time we can tell when this has happened, and we have ways of compensating for it. It's not always as easy as in those pictures of basically flat layers that I showed you earlier. There are even a few cases where you can have pieces of the Earth's crust completely overturned, so the youngest end up on the bottom and the oldest is on the top, but that's rare. And when it happens, we can generally tell. You don't scramble rock layers like that without leaving a lot of physical evidence that geologists can find and look for. Okay, so how am I doing time-wise, folks?
You got about 10 minutes.
Okay, great. I have time to talk about William Smith. Started out his career in comedies like The Fresh Prince of Bel-Air, and released an album in the 80s with DJ Jazzy Jeff. Really made his big breakthrough role in Men in Black, but he's gone on to play more serious roles like 7 Pounds and Ali. Wait, sorry, wrong William Smith. This William Smith was a British engineer and surveyor, heavily involved in digging canals. At the time when he was working, the late 1700s and early 1800s, Great Britain was going through the early phases of the Industrial Revolution. And one of the things that was necessary was a way to move stuff around. Railroads had not yet been built, although they would be soon. And, there's no cars, no trucks, no modern highways. So a network of canals was built, and it's still in operation all over England. And it was built so that you could get large amounts of raw materials like coal or iron or whatever was needed to the factories, and then get the factories' products to the places where they'd be sold. So this is an early infrastructure project. Smith was involved in surveying a lot of these canals, planning the routes. And because he was always helping build these canals, he saw more rock layers than anybody else. Rocks are usually covered up with soil and plants and things like that. But just as now you have to blast through rock layers to make roads, you had to blast through rock layers to make canals. And Smith knew more about the rock layers than anybody because he was blasting these canals. And he collected fossils from these rock layers. They're all sedimentary in that part of the world. He knew that the layers followed the basic rule of older ones are below, younger ones are on top. He noticed that different rock layers contained different fossils. And what's more, fossils appeared in a predictable order of time. The order of fossils, if you worked it out for one place, you would find fossils in the same order all over England, and in fact, all over Europe. This came to be called Smith's law of succession, and let me just show you. Back in the days when I did quite a lot more traveling than I do now, I spent a lot of time in the Mojave Desert of Western North America. Southern Nevada, southeastern California. And if you go digging in some of those rock layers, you find fossils called trilobites. If you saw one, you might think it was a roly-poly bug or a shrimp or something like that. They have segmented bodies, lots of little legs. They lived in the ocean, did not come out on land. They had lots of legs and crept around, or sometimes they swam. They have died out. We have not had any trilobites on Earth for a very long time. But you can dig up lots of fossils of them if you know where to look in the Mojave Desert, in the California-Nevada border area. And if you dig in rocks that I used to dig in, a set of layers called the Wood Canyon Formation, exposed in the Spring Mountains north of Las Vegas around the ghost town of Johnny, you'll find a genus of trilobite called Nevadia, which is illustrated down below. If you go up in the stack, you don't find a lot of fossils of any kind in the rock layer above the Wood Canyon. It's called the Zabriskie quartzite. But if you dig in the bottom of the layers on top of that, called the Carrara Formation, you encounter trilobites that are classified in a different genus called Olinellus. They look pretty much like Nevadia, but there are consistent differences, and you could learn to tell them apart with a little practice. Climb up higher in the Carrara Formation, and you'll find trilobites of a completely different type in the genus Cispocephalus.
There's 5 minutes.
Okay, who cares? Well, if you find these fossils anywhere in North America, they will be found in this order of appearance. You could go to certain parts of Georgia and dig up trilobites. There's places you can do that. And if you find them, you'll find them in this order. Nevadia is always in rocks below the rocks where you find Olanellus. And Olanellus is always in rocks below the rocks where you find Cispocephalus. If you were to go to, there's parts of British Columbia where you'd find these. You could do the same thing in Siberia. You could do the same thing in eastern Newfoundland, in Canada. Sorry, western Newfoundland, in Canada. Now, much of the world, you would not find these three at all. They would have other fossils completely. But if you find these anywhere, you always find Nevadia as the older of the three, Olanellus in the middle, and Cispocephalus up on top. It's predictable. What that lets us do is define periods of time in Earth's history based on what was living at various times. It can get a lot more complicated than this, but to give you an example you've heard of, you may know there was a time when the dominant land animals were dinosaurs. So here's some dinosaurs running around what is now Hempstead County, Arkansas, about 100 million years ago. And the dinosaurs lived, and then, except for a little group of dinosaurs called birds, they died out. They disappeared. We ain't got them no more. And after that time, the dominant land animals come to be mammals. So we can define a time period, which we could call the age of dinosaurs. And after that time, after the dinosaurs are no longer here, we have a time period that's younger, which we could call the age of mammals. Now, we'd actually subdivide that age of dinosaurs into sub-time periods. One's called the Triassic, one's the Jurassic, and then the latest would be the Cretaceous. And we'd subdivide that age of mammals a lot more finely as well. But I'm just trying to give you the flavor here that we can use the sets of fossils that are living in a particular order to define time periods in the past. Just like if we were looking at US history, we could define a certain time period to be the age of steam, because that's when people built steam engines. And then after that came, I don't know, the age of gasoline, because people built gas engines and quit building steam engines. And then after that comes the computer age, because now we all use computers and don't go anywhere. But just as you could define periods in human history by things that people were using, you could define periods of Earth's history by the identities of the animals and plants that were on the planet at the time. And by 1850, scientists had worked it out. Life on Earth has a history, probably an extremely long history, although nobody knew exactly how long just then. Here's some evidence for that history. That's dinosaur bones left in place for people to see at Dinosaur National Monument outside Vernal, Utah. Well worth a visit if you're ever out that way. Life's got a history, and you cannot fully understand life today without knowing something about its history. Just like you can't understand why the United States is the way it is without knowing how we got this way, you can't fully understand why life on Earth is the way it is without knowing how it got this way.